represents a digitized microscopic tissue that is used to detect cancerous metastasis in order to evaluate the extent of the breast cancer, and comes from a Chameleon 2016 competition, where image as well as lesion level labels were provided. So such metastasis, for example, represents a visual anomaly, and pathologists need to inspect such tissue samples, usually under the microscope, not only to detect such regions on a slide or lesion level, but also to assess different prognostic markers, to stage the cancer, and to come up with an appropriate treatment plan. So with the availability of whole slide scanners, such tissue samples can be digitized with a resolution of up to a few hundred nanometers per pixel, which results in such gigapixel imagery on which we can apply automated methods. So the raw histology imagery needs to be pre-processed, which usually means extracting the actual tissue and then extracting the patches from the tissue, because the whole tissue cannot be directly processed due to its huge size. So in this case, we extract all the patches which have more than 90% of the tissue coverage. And in the cancerous patches, we also make sure that at least 90% of the patch is covered with cancerous tissue. For the training set, as I will mention later on, we only use healthy patches, while to test the methods, we use both classes. So most of the current problems are approached in a supervised fashion, including in the medical images analysis domain. So here we focus on histology imagery, but the approach is similar for all the modalities. So this, for example, represents a winning approach from Chameleon 2016 competition, which similarly extracted patches from whole slide image. They labeled them accordingly to the provided annotations, so of either being normal or tumorous. These then presented an input to the deep learning architecture, GoogleNet in this case, such that a patch-based classifier was learned, which was then applied to overlapping patches to produce a heat map. This heat map was then used to localize the actual metastasis. So in this competition data, the method achieved pathologist-level performance on both slide as well as lesion level detection tasks, and those results were further improved in later works. So the real problem lies in obtaining label data, which is especially difficult for such biomedical applications, where only trained domain experts can provide such labels. And the data itself is usually also of high complexity in terms of representation as well as dimensionality. So in an unsupervised case, we only have access to normal, in this case, healthy samples, which are used to normal the normal appearance. And anomalies are then detected as deviations from that learned normal visual appearance. So this is usually implemented with autoencoders or GANs. So for example, in autoencoders presented in the left-hand side, we have a direct reconstruction. So basically we autoencode healthy samples, and then anomalous samples cannot be well reconstructed. And thus we are able to detect anomalies from the differences. In reality, autoencoders have this memorization effect, and they tend to reconstruct also unseen anomalous samples quite well. So the other approach are GAN-based approaches, where you similarly model the normal appearance, but with a GAN networks. In comparison with autoencoders, GANs do not automatically yield this inverse mapping into the latent space. So different optimization techniques were proposed to find the optimal latent vector representation that would generate the closest looking normal appearance samples to the presented query image, which you don't know if it's cancerous or not. And then we similarly detect deviations based on some thresholded visual similarity difference. So the clear example of such methods that was recently published is ANOGAN method, which is one of the first methods that implements this paradigm. So unsupervised anomaly detection approaches are not well researched in the medical domain. So we have already explored in the last year MICAI conference, high resolution histology imagery synthesis for normal appearance modeling. So this is basically this part of the ANOGAN framework. There was also just this year at ISB conference, they presented a method which basically they also adapted this second part to implement the whole pipeline. And concurrently to this approach, we present a complementary approach that approach the same problem in a different manner that I'll present now. So our approach is based on image-to-image translation, which is a GAN-based framework that learns a mapping between different domains. So in a supervised fashion, where you have pixel level correspondences. In an unsupervised fashion, you only have images that are not paired at all. So in this work, we show that image-to-image translation alone can be very effective for unsupervised detection of visual anomalies in histology imagery by using a recent image-to-image unsupervised approach that disentangles content that needs to be preserved apart from style during this translation process, which is particularly suitable for digital pathology. So we achieved that by pre-training. Basically, we performed image-to-image translation just on healthy samples with which we effectively learned normal visual representation. And then we performed example-guided image translation, which was unable to fully reconstruct cancerous patches. In such patches, there was we can even see that it showed some level of artificial healing on them, while, for example, normal healthy samples were reconstructed much more accurately. So if we look at the results that we have obtained for such an unsupervised anomaly detection approach, so we performed the experiments on the chameleon dataset. We pre-trained the model just on healthy samples, and then evaluated the reconstruction ability on healthy and cancer samples with different distance matrices, and the reconstruction distribution, error distribution, or let's call it anomaly score for those different matrices is presented in the graphs below. So we can see that the two distributions are quite well separated with the proposed method, while, for example, Anogan fails to sufficiently separate the two distributions. This can be also seen from receiver operating curves and associated AUC scores that were computed at Uden index, where we have also compared our approach against the supervised baseline on the patch level. And we can see that similar performance was achieved just with healthy samples seen during the training without any additional labels. On the right-hand side, we can also see the results from the just published concurrent work that I mentioned, which basically implemented the Anogan idea. They exchanged the vanilla GAN network that was used in the original work with a more sophisticated one. And they also proposed a new distance matrix that was based on Kenny edges. So we can see that the baselines are very similar in distributions as well as AUC score, but overall, we can see that despite we cannot directly compare the approaches because they have used the same data, but they sampled it differently, we can see that our approach significantly surpasses it. They also didn't make any comparison with a supervised counterparts. So here we present also example reconstruction of healthy and cancer tissue samples for all the methods. So we can see that the proposed method reconstructs healthy samples much more accurately in comparison with, for example, StyleGAN2 method. So some level of artificial healing is visible on cancer samples, we can see visual appearance much more closely resembling healthy samples. The F-Anogan method, for example, it's only able to operate on 64 by 64 patches, where we similarly notice that better reconstructions were obtained on healthy appearance samples. So the visual patterns in the StyleGAN2 method reconstruction demonstrate significantly lower variability in the visual appearance, especially in comparison with its demonstrated capability to synthesize really realistically looking samples. So in comparison, our proposed image-to-image translation method enables high-resolution image synthesis, as well as example-based reconstruction that can be effectively utilized for the detection of such visual anomalies. So to conclude, we presented an unsupervised base approach for visual anomaly detection in histology imagery that significantly surpassed current unsupervised approaches, as well as almost reached the performance of the supervised counterparts. So we think that this concept is very applicable also for general pre-training, especially in medical imagery domain, where you have, for example, different stains of the tissue, different magnifications, different contrasts, for example, that are used in MRI scans, that can be all much better utilized in a more meaningful meta-learning tasks in comparison with current self-supervised approaches. So in part of our future work, we also want to extend this framework with task-specific heads that can utilize such pre-trained representation to fine-tune on specific tasks where you have also limited fully labeled data available. And one of the future research directions is also to get an answer on how such concept generalizes to other domains. So the problem that I presented, the tumor region detection, is the problem that it's quite well approached in the literature, but it's also the problem that it's the least interest to the pathologists to be automated, because for a seasoned pathologist, this kind of problems are quickly solved. What's the real, let's say, bottleneck for the pathologist is, as already Jens mentioned, is the analysis of the immunohistochemically stained slides. So for example, instead of detecting tumorous regions, they also need to quantify it on a cellular level. What are the positive and quantify the positive stains, positive cells and negative cells, quantify them. This basically involves a lot of manual effort, counting thousands of cells. And this is basically the next problem where I see the most proliferation of the methods that are less data intensive, because also the result that is expected has also much more important clinical value. So this is the problem that we're currently addressing with our clinical partners, and I hope that I will be able to present it to you in the near future. That would be all from my side, and I'm open for questions. Thank you.
Well, thank you very much, Dejan, for a very nice talk. So we have time for one or two questions.
Hi, can I ask a question?
Yeah. Yes, please.
Hi, this was great work. I was wondering, how does this generalize to the task of not just telling healthy tissue from cancer, but actually from being able to tell one, let's say, subclass from another, the grading question, where the differences are more subtle, but they result in different grade and different treatment?
Yes, we currently only focused on the one class problem. So basically determining if the tissue is tumorous or not, so cancerous or not. So we are currently exploring basically to translating this problem into these neuroendocrine tumors that I'm showing you now. So in this case, we'll have 3 different classes of cells. So we'll be able to determine also the ability of such methods to differentiate multiple classes. I hope that I answered partially your question.
Yeah, Dejan, given I have a lot of experience in this area, so what you could do in the future, you could try to find densities of cells, cancers, and the different variations, so you can possibly, with neural nets, determine different types of cancer, or mutations of the cancer, or different types of cancer within these slides. So it's possible. So I think that was the question about, and I think with what you've been doing, I think you could extend it to fine-tuning your analysis of cancer.
Yes, so the main idea, the main idea was basically, as we see in the medical image analysis domain, all the approaches that are out there, at least up to this year, they were mostly really based on supervised approaches. So we take this base approach that was approached by other researchers in a supervised fashion and try to see if unsupervised approaches are really even appropriate in this domain. Now we also envision, yes, lots of further tasks to deploy such methods.
Yeah, very good. So I think in the interest of time, we'll have to move to the next talk. So the next paper is titled RNN-SliceNet, a slice and cluster approach for nuclei centroid detection in 3-dimensional fluorescence microscopy images. And Liming Yu from Purdue University will present.
All right, thank you. Can you hear my voice? Okay.
Yes.
Oh, okay, great. Hello, everyone. My name is Liming Wu. I will be presenting our work with the RNN-SliceNet, a slice and cluster approach for nuclear centroid detection in 3-dimensional fluorescence microscopy images. And this work was developed at the Video and Image Processing Lab at Purdue University. So to start with, I'll briefly talk about the introduction and motivation of our work. So fluorescence microscopy is an optical microscopy that images the subcellular structures of living cells and animals. For our case, we use a 2-photon microscopy to collect the data from the kidney of the living rats. And recent advances of fluorescence microscopy enable the acquisition of a larger amount of 3D volumes with a much higher image quality. Robust and accurate nuclear centroid detection is an important step for understanding of these biological structures in these fluorescence images. However, we are currently facing several challenges. For example, most object detection methods work only on 2D images and are difficult to extend to 3D volumes, such as the regional convolutional neural network-based method. Also, segmentation-based models, such as 3D UNet or VNet, these models can be used on 3D volumes, but they are generally more computationally expensive and have difficulty distinguishing different instances of objects. Because these methods are designed for semantic segmentation, so post-processing steps are required to separate different objects. And also, deep learning-based models typically require a large amount of training samples, and manually annotating this ground truth is labor-intensive, especially for 3D microscopy volume. So to address these issues, we present a slice and cluster strategy to detect nuclear centroids in 3D microscopy volumes using synthetic data generated from SP cyclogram. For our data set, we use four types of data denoted as a data 1 to data 4. Data 1 consists of one volume of size 128 by 128 by 64 pixels, and data 2 consists of 16 volumes of a similar size. Data 3 consists of 21 volumes of similar size, and data 4 consists of 90 volumes of larger size. Data 1 to data 3, these are obtained using a 2-photon microscopy from rat kidney. But for data 4, it's actually from the public collection. So for our proposed method, as shown in figure 1, our method consists of 3 main modules. The first module is a 3D synthetic data generation. In this part, we first generate the synthetic nuclei segmentation masks. This is actually a 3D volume, 3D binary volume that contains the segmentation mask. In order to generate this mask, we model the nuclei as ellipsoids with a different rotation and size. So we can randomly generate these binary nuclei and put them in the random location of an empty volume. And we can also control the density of these nuclei. And along with original microscopy volumes, we can train SP cyclogram. And for the trained SP cyclogram, we inference it on some other new binary volumes to generate synthetic microscopy volumes, which look like the original one. And then we train our RNN-SliceNet detector to detect these nuclei from different directions. Actually, we inference it on this detector on 3 different directions of a volume, and to generate 2D detection centroids on each slice of the image. And then we use a clustering method to cluster these 2D detection centroids and then to find the cluster centers. We use that as a nuclei, 3D nuclei centroids. And finally, we use a 3-way majority voting to fuse these results from 3 different directions. Okay, so for 3D synthetic volume generation part, we based on the observation of the original microscopy images, we can model the nuclei as an ellipsoidal shape with a random rotation and location in a volume. Then after we generating these binary volumes, we use SP cyclogram to train to translate these binary volumes to synthetic microscopy volumes. The SP cyclogram basically is an extension of cyclogram. So it consists of 2 generators, 2 discriminators, and a segmenter edge. And this edge can preserve the spatial location of the nuclei. So this is very different from the cyclogram. And there's an extra loss function called spatially constrained loss in SP cyclogram. So this is actually a L2 norm, can be used to preserve the spatial location of the nuclei. As shown in figure 2, this is an example of generated synthetic image slices. The first column is our like 2 original image slices from data 2 and data 3. And the second column are the synthetic microscopy images generated from the third column. During our training, so the second column and the third column will be used as the training samples to train our RNN-SliceNet detector. Our detector is actually based on a modified version of faster RCN. We use a VGG16 as a feature extractor to extract features from the input image. And the RPN module, the region proposal network, will be used to provide the candidate regions of the image. So that can indicate where there might be objects. And these candidate regions are sent to ROI head network, which is the last part. Then this RI pool module will crop the features on the extracted feature map based on these candidate regions. And then these cropped features are sent for regression and classification and to output the where the centroid is. And for the figure 4, it shows the overview of the how we inference our detector on this 3D data. We basically inference our since RNN-SliceNet is work on 2D, so we inference it on 3 different directions of a volume to generate the 2D detection centroids on each slice. These are in the red dots you can see here. And we use then we use a hierarchical clustering method to cluster these 2D detection centroids and to output the 3D centroids, which is in the red color here. And finally, we use an extra hierarchical clustering model to fuse these results from 3 different directions. Okay, so for hierarchical clustering, we specifically we use a 3D agglomerative hierarchical clustering with average linkage criterion. And then we use the Salama coefficient to determine the optimal number of clusters in a volume. So in order to estimate the Salama coefficient, we first estimate the mean intra-cluster distance and the average of the mean inter-cluster distance, as shown in this equation. So for each point, we estimate this distance, and then we can use this equation to estimate the Salama coefficient for each cluster number. And then the optimal number of clusters is associated with the highest Salama coefficient. And then we use that as the number of nuclei in a volume. For the experimental results, we can see in fact figure 5. These are some examples detections from RNN-SliceNet on several slices of the 4 different data. Figure 6 shows a 3D visualization of nuclear centroid estimation. We compared with several different deep learning based or segmentation based models, like 3D UNet, VNet, and DeepSense. So we can see here the red dots are estimated centroids, the green dots are ground truth centroids, and the yellow is the overlay of green and red. Maybe these are very small to see, but let's take a look of the second row. We can see our method is on the last column. We can see it captured almost all of these objects, even the partially included nuclei, and it has a better overlay with the ground truth because there are more yellow color there. That's for the visualization, and for the statistical evaluation results, we use the mean absolute percentage error, the MAPE, to estimate the counting accuracy, and we use the mean average precision to evaluate the centroid detection accuracy. We evaluated on 4 different data. For data 1 and data 2, we train our model on synthetic data only, and then tested on the real data. But for data 3, we adopted the idea of transfer learning. So we first pre-train our model on the synthetic microscopy data 3, and we updated our model on several volumes of real microscopy data 3. And finally, we evaluated on the rest of the real data 3. For data 4, since this is from public collection, so it's very relatively easier. So we did not train our model on either synthetic data 4 or real data 4. We just use a model from the data 2, then we inference it on the data 4. We can see our method achieves the best accuracy on the centroid estimation and counting. To conclude, we present RNN-SliceNet using the slice and the cluster strategy to detect 3D nuclear centroids. I think our idea is very straightforward because we just first borrow other popular 2D detection network, and then we run it on 3 different directions of a volume, and then we figure out a way to fuse these results. So our network is trained on synthetic microscopy volume interface. In the future, we can extend generate for training RNN-SliceNet. So I guess that's pretty much of my presentation. I'll leave the time for the audience if there are any questions.
Thank you very much. If somebody has a question, we have time for one question. You can ask directly, Alain.
Hi, I'll ask a simple question. So for your original images, the real images, how did you get the ground truth?
For the original image, we manually annotated this ground truth. So these ground truths are very limited.
Yeah, let me also insert something here. The images were then verified by a biologist. So they helped us with the ground truth.
Mm-hmm. Were there a comparison done where, what if you just trained on the original images without using synthetic data?
The problem is we didn't have enough of the original images, ground truth, to be able to do that.
I see.
We only have several volumes.
Yeah,
So
we only have a very few volumes because they're very, very difficult to ground truth.
Mm-hmm. Is this the first time where a 3D model for generating this kind of synthetic data being tested?
No, we've been actually looking at these types of synthetic images since 2017.
I see.
We presented a paper in 2017 at this workshop where we introduced this idea of the 3D SP cyclogram.
I see.
For generating synthetic images. You can go to our website and see all the papers we've done on this, on these synthetic images.
I see. Thank you. Thanks for the answers.
All right, thank you very much for the very nice talk. So we move now to the next speaker, who's going to present the paper Snapshot Deep Learning Approach for Instant Segmentation of Research Imaging Data, State-of-the-Art Cloud Service, DLGram, and Alexei Kenneth from Novosibirsk State University.
Thank you very much. I hope you see my presentation.
Yeah? So far I see you. Maybe share your screen. So.
Okay, I'm sorry.
Yeah.
Is it okay?
Not yet. Oh, no? Yeah, now I can see, yes.
Yeah. Thank you for opportunity to make a presentation.